Tonight is our first two graduate speakers, and Heather and I were just talking right before the meeting how these are our favorite lectures, even though some of it goes a little over our head. There's just so much interesting research being done out there, especially at the local colleges, and we're super proud to support it and to hear from them. So tonight we have José Solórzano and Courtney Meeks. Jose is going to go first, and I'm going to just share a little bit about him. He is a PhD candidate at the University of Minnesota. He's researching the biology, pathogenicity, and epidemiology of, I'm going to try to say it right here, Phyllachora maydis, which is the invasive fungus responsible for the economically significant tar spot disease in corn. A recent member of the Minnesota Mycological Society, he's also an active member of the Mycological Society of America, where he led the creation of and currently leads the organization called Spores, a mentoring program for undergraduate students. He's also a member of the Mycology Club at the University of Minnesota and participates in journal club discussions and extracurricular activities. So welcome, Jose. Thank you for being here to present to us tonight.
Yeah, thank you so much for the introduction. Thanks for inviting me, and it's such an honor to receive the student scholarship. And I'm also happy to be a member of the Minnesota Mycological Society. I've been a member of the the Mycological Society of America for a long time, and I really wanted to be involved with this local society a little bit more. So I'm really happy to be here today. So part of my research, I work on tar spot of corn, as Kathy mentioned. Tar spot is a really important and economic important disease in the continental US. And then specifically today, I'm going to talk about one of the projects that we have been developing in the past couple of years to identify the diversity of Phyllachora species that infect corn and also identify their host range. So we do this because we want to better understand what the tar spot disease and also develop management strategies and control and prevention strategies for this disease. So in my talk today, I'll talk about a little bit about the history of the disease, the history of the fungus, Phyllachora maydis, and also I'm going to discuss the activities that we are conducting to identify these species and their host range. There are also two morels in my presentation. I know this is the morel season, so pay attention and see if any of you can find the two morels that are in one of my slides. So I'm gonna start with the history of Phyllachora maydis. So Phyllachora maydis was first found in Mexico in 1904. It was found infecting corn, and then the Phyllachora species was named after corn. Phyllachora maydis, a pathogen of corn. So after being found in Mexico, this Phyllachora maydis pathogen was reported in many other countries in South America, Central America, and also in the Caribbean. But it was not until 2015 that the disease was found in the continental US. It was reported from Indiana and Illinois, and after these, the first report of the disease was made in 2016. After being found in Indiana and Illinois, the disease was also reported in many other states in the Midwest. And in 2019, the disease was found in Minnesota. And in 2020, the first report of this fungus was made or published in 2020. So on the right, you can see the map of the geographic distribution of Phyllachora maydis in the continental US, where orange represents the places where it was found in 2024. And the gray color counties represent the places where Phyllachora maydis was found prior to 2024 and after 2015. So the fungus is pretty much found in the Midwest area of the country, but it's also found in a few states in the South, namely Florida and Georgia. And in general, so this fungus represents a big threat for corn production in the continental US. So it is very important that we study this pathogen because it is considered an invasive species in the continental US. So Phyllachora maydis, this fungus, when it infects corn, it produces tar spots. So tar spots is the name that we give to the stromata produced by the fungus. Stromata is a technical term used to describe tar spots. And these are the tar spots, the black spots that you see on this corn foliage. So Phyllachora maydis infects the leaves, the husks, and also the stalks of corn plants. And they initially start as these small, rounded black spots, as you can see on the left. And as they develop, they become elongated and they grow with the leaf venation, the leaf veins. So these tar spots are embedded in the leaf tissue. So if you try to remove them with your finger, you wouldn't be able to do that because they are attached to the leaves. So inside of these tar spots, the fungus produces fruiting bodies, namely Perithecea and Pycnidia, which bear ascospores and conidia respectively. And these are very key important diagnosis features for these Phyllachora maydis pathogen. And it is not just a thing that is produced by Phyllachora maydis. Most Phyllachora species produce tar spots. So these are key characteristic features for these Phyllachora species. So in my research, in my journey to understand tar spot of corn and to understand the environmental conditions required by this fungus to produce this disease, and also understand how this pathogen infects corn and if it has the capacity to infect other hosts or other plants, we developed inoculation methods in my lab here at the University of Minnesota. So we developed two different types of inoculation methods. And what I mean by inoculation methods, this means methods where we use spores of a fungus to infect corn plants. So there is one thing I didn't mention before about Phyllachora maydis. Phyllachora maydis is an obligate biotroph organism. What that means is that we cannot take this fungus to the lab and grow it in culture media. This fungus only grows in corn and only reproduces in corn. So working with this fungus is a hassle in a few terms. So we developed these inoculation methods to understand how this fungus infects the corn plants. And in these two methods one is for control environments and the other one is to understand and study this fungus in the field. So understand how the fungus spreads from plant to plant. But from these inoculation methods, we learned that the fungus Phyllachora maydis survives or overwinters in corn residue. And that the spores that are left behind in this corn residue can infect a susceptible corn plant when the environmental conditions are conducive for the disease to develop. And when that happens, the disease will show up and the tar spots will appear. But for the emphasis of this presentation, we will focus on the survival of the pathogen and the inoculant source here on the left. Because we know that the fungus can survive in the corn residue. However, it is hypothesized that corn residue might not be the only source of inoculant during the non-growing season. It is considered that there might be other ways, in other places where this fungus may survive during the non-growing season. So that brings us to a study published in 2022. It is a study that says that there are many other Phyllachora species that may be associated with corn or may be responsible for corn tar spot. So this study that was conducted by Brothers et al., or Brothers and many other scientists, they collected contemporary and historical samples of different Phyllachora species, and they included Phyllachora maydis, the corn tar spot. And so here's a picture on the right, and on the top panel, they have examples of these Phyllachora collected from corn, and also they have samples, pictures on the bottom of different Phyllachora collected from grasses. I know these are small pictures, but you may see the black spots on these grasses, and those are the tar spots that they used to collect this fungus for the next experiments that they conducted. I'm going to show you more pictures of these in other slides. So they conducted this study to understand if it was true that there were other Phyllachora species that may be associated with corn. From these analyses, they collected all these historical and contemporary samples from grasses and from corn. And they sequenced the ITS and LSU regions. These are two genes from the genome of Phyllachora. And I know and from these, they built or conducted an analysis called maximum likelihood, and they generated a tree. I know this is blurry on the left, but you don't have to worry about that right now. So they conducted a maximum likelihood analysis to observe the relationship or how these species are related. From here, they identified that Phyllachora collected from corn and those collected from grasses represent a complex and closely related species. So if we zoom in this tree, in one of the clusters or one of the groups from this tree, we observe that there are different Phyllachora species from different plant hosts from different locations. So the black color font represents the different Phyllachora species. The blue color font represents the different plant hosts, and the red color represents the different locations from where these Phyllachora samples were collected. So in this cluster, you are observing different Phyllachora species, including Phyllachora maydis. You see right here in the center of this group, there are different Phyllachora species, Phyllacula vulgata, and other Phyllachora species collected from different grasses, from different locations. But most importantly here, they have Phyllachora maydis collected from corn in the contemporary range of tar spot of corn. Michigan, Minnesota, for example, which is important, Iowa, Wisconsin, and Illinois. So Brothers et al. Say that there might be more than one Phyllachora species associated with corn, and based on this analysis, they indicated that there might be 5 to 3 Phyllachora species associated with corn. And they limited them by the clusters that were formed from this analysis. You see the 5 clusters here, 1, 2, 3, 4, and 5, and they name it from 1 to 3, and then 4 and 5. So what is the take-home message from here is that with this information, the authors inferred that these species may also infect corn, these other Phyllachora species, but also that Phyllachora maydis may have the capacity of infecting grasses, native grasses in Minnesota, for example, and any other states in the Midwest. So doing my literature review analysis, I found a paper from 1989. This was written or was conducted by Gavel in 1989. And in this article or analysis conducted in the Midwest in the US, they collected multiple native grasses and they found multiple Phyllachora species associated with these grasses. However, they didn't find Phyllachora maydis. They just found a lot of Phyllachora species. So we know that there are different Phyllachora species that are present in the Midwest and that these Phyllachora species infect those grasses. So this is some part of the background for you to understand that there are Phyllachora species found in the US that naturally live here in the US. And now we have this invasive Phyllachora maydis that just emerged in the US in 2015. So now with my research, I've been working on tar spot of corn for the past 4 years, for 3 years and 9 months. And while I've been working on this, I've been collecting samples across Minnesota, and also other collaborators have been helping me to collect samples. So while I've been doing this, I've found grasses with tar spots in next to cornfields. I have also found samples or grasses with tar spots in non-agricultural settings in Minnesota. But what I mean by this is a place that is far away from a corn farm. And I have also found grasses inside of cornfields or in between cornfields with tar spots. So this raises the question, can Phyllachora maydis infect these grasses? And the other question that it raises is, can other Phyllachora species infect corn? So to understand or to answer these questions, we are conducting a survey-based analysis to ID alternative hosts and other Phyllachora species that may be associated with corn. So the study that was conducted by Brothers et al. That was published in 2022, it was just molecular analysis. So my work here will be molecular, the first part, and we're gonna conduct this sampling across the Midwest. We have different collaborators from different universities that are helping us collect the samples to capture the diversity of species that may be present or that may be affecting corn. So just 10 days ago, I was conducting a sampling close to the St. Croix River, close to Wisconsin, and I found a few grasses that had tar spots on them. And this is a normal finding for me because I found these grasses before, some of these grasses in other places. But you can see here in these grasses that they have the tar spots in them. If you take a closer look to them, you will see that they have these black spots on them. And one of the things that we do to make sure that these are tar spots, we try to pick them with our fingers and make sure that they don't drop off or get removed from the leaves, because we know that tar spots should be attached to the leaves or embedded to the leaf tissue of these plants. So these are mostly Elymus species. These are common native species in the state of Minnesota, but these are also found across different other states in the Midwest. So I collected these ones, which is important for me because we're trying to make sure that we are finding those plants that may serve as reservoirs of a corn pathogen. And from here, we're going to conduct more sequencing or molecular analysis. So we take these to the lab and we rupture them, we open them up, we analyze them in the microscope and make sure that they have ascospores. So if you see the structures right here on the top, on the lower corner, right corner of your screen, you see these banana-shaped structures. These are ascus or aci in plural, and they have ascospores inside. So for us, this is a first confirmation that these might be Phyllachora species. And this is before we do the molecular analysis. So this is also another example, another leaf that I found in the field, and this was like just laying on the ground. So we collected all the samples, and with collaboration from many other scientists across the Midwest, we have a database of Phyllachora species collected from grasses and Phyllachora collected from corn that contains around 270 samples now. And from these, we would like to present some preliminary results. And from this survey-based analysis, we conduct a molecular analysis, which is a phylogenetic tree, a phylogenetic maximum likelihood analysis. We have identified that Phyllachora maydis is present in corn only, and also that Phyllachora species found in grasses had not been detected in corn. So more work is being done, and I'm conducting more work to identify these Phyllachora species, and also we are sequencing more genes to better clarify these questions that we have. So the next step in my research is to conduct inoculations using the inoculation methods that we develop in my lab. So and for these, we are selecting a set of different plant species that are genetically different, but that are also found in the continental US and that are also found in the native range of Phyllachora maydis, for example, Mexico and Central America. So for these, we selected a few corn relatives from Mexico and Central America, and we are selecting these because the chances of Phyllachora maydis of infecting these corn relatives may be high, because Phyllachora maydis is also native to Mexico, so the chances of these pathogen or fungus of infecting these plant species is very high. And we're also collecting or testing these Phyllachora maydis in native plant species that are known hosts of Phyllachora maydis from the Midwest. And so we have a pool of 20 plant species, and from these we're conducting we're developing experimental populations. We are analyzing those in the field, but also in greenhouse conditions. So right now in the summer, we'll be conducting these or assessing these host range in 3 different locations. And last year, we analyzed this in 2 different locations. So the 20 plant species that we selected were based on the plants that are known hosts of different Phyllachora species based on the work conducted by Gavel in 1989. And also some of the plants come from the genera mentioned in Brothers et al. 2022, the recent study. And the rest of the plants are corn relatives. So this is me. Here's a picture of me collecting or conducting sampling in the field last year to assess the host range of the corn pathogen. So for these, we have some preliminary results, and And those are that the different corn relatives are hosts of the corn pathogen. So these are grass plants, but these are corn relatives, really close to corn. So we tested all these plants in the field and also in the greenhouse or greenhouse experiments, and we were able to observe the development of tar spot in these plant species. And if you are able to see these images, you will see that they all have tar spots. They all have tiny little spots, and this is just 12 to 13 days after we inoculated them with the pathogen. And you will be able to see the tar spots here, the little black spots on all of them. So these are 7 different taxa. These all come from this genus Sia. Sia is the genus of corn also. So this for us is a very important breakthrough in the understanding of tar spot of corn and also understanding the biology of Phyllachora maydis. And this is very important for us because we know that the fungus that infects corn has the capacity of infecting other plants that are not corn. While we don't have these plant species growing naturally in the continental US, these may play important roles in the development or the spread of this pathogen in Mexico and in Central America, for example. So with this information, we are better understanding the disease cycle and the life cycle of the disease cycle of tar spot and the life cycle of Phyllachora maydis. And what are the things that we're learning about this, about the tar spot disease cycle? We're learning that the fungus could survive or overwinter, well, survive during the non-growing season in these corn relatives. These are grass hosts that can serve as alternative hosts of this pathogen. And they can serve as inoculum reservoirs during the non-growing season that can later infect more susceptible corn during the growing season. But also, the spores that may come from these grasses may be able to infect again these grasses and continue the cycle of this pathogen and helping it survive for longer periods of time and stay active for infection. So, in general terms, what are the summary of these research, the preliminary results, the summary of the preliminary results that we have? Phyllachora maydis infects wild grasses that are correlatives. So this means that Phyllachora has the potential to infect plants that are plants other than corn, and these grasses can serve as alternative hosts in Mexico and Central America. Phyllachora maydis has been detected in corn only, but not in infected grasses in the US. So grasses unrelated to corn may not play a role in the tar spot disease cycle, but more work is being conducted to clarify this. And the other conclusion here is that Phyllachora species found in grasses have not been detected in corn. And so the implication of this is that other Phyllachora species we have identified on grasses might not pose a threat to corn. But this is just preliminary data, so more genes are being sequenced to limit species in a better way. And this will help us understand better the biology of this pathogen and also the tar spot disease cycle. So with that, I would like to thank all the people that have, the collaborators and people that have worked with me. I also want to thank the Minnesota Department of Natural Resources for providing us a permit to collect samples in the state of Minnesota. The USA ARS National Plant Germplasm System for providing us with seed for some of the host range analysis, and the Minnesota Invasive Terrestrial and Plants and Pest Center for funding my research. And thank you all for inviting me, and also thank you very much for the student scholarship, and I really appreciate it. It's going to be really important for the research that I'm conducting. And I'll be happy to answer any questions, and I hope somebody was able to find them or else in my presentation. Thank you.
Thank you. We found them. We saw them.
Great.
Did you pick them and eat them?
I did.
Good. I had a question about I mean, I think it's maybe obvious, but because it's affecting the tar spot is affecting the leaves,
Mhm.
does it make the plant less able to produce the corn cobs? Does it just affect the growth? because the tar spot doesn't actually affect the corn cob, just the leaf, right? But then does that tar spot, I know lots of times they make silage out of corn too
Mhm.
to feed animals. Is it toxic to animals if it's infected?
That's a really good question, and a lot of people have been asking us about these for a long time, and recently a paper was published about silage and how tar spot may affect silage and the production of that. I can tell you something about this. So sometimes we see these tar spots. We only see like a few spots on a leaf, but sometimes we see like 75% coverage of the leaves or 50%, 25%, and that's a lot of coverage of the leaves. So in terms of photosynthesis, these tar spots may affect the natural, capture of energy by the plant and also the light. So the transformation of this energy may affect the production. And yes, there are studies that were published, for example, in 2018, where they mentioned that the presence of this disease has affected significantly or in a big way corn production. So the yield has been affected largely by the presence of this pathogen in corn plants. Um, we don't know exactly the fungus process toxins, and I think that will be a great thing to to research and study. I can tell you that the fungus is an obligate biotroph organism. So usually this fungi biologically try to keep the cells that are around them alive for a prolonged period of time. So the plant keeps feeding them and providing them with nutrients so they can survive until they reproduce. Um, and that may contradict the production of toxins right away that may be toxic to other pathogens or to people, for example, or to humans. Um, But that will be a great thing to investigate and see if they pose a threat to humans or to animals. Um, because I know like in Minnesota, corn plants are used for feeding, for animal feeding, and for many other things, and that'll be a major thing. Thus far, we haven't heard of anyone complaining about intoxication of their cows, for example, or anything like that. Um, but it will be something really interesting to investigate in the future.
Thank you. Howard, you have a question too. It's in the chat. Is there a prognosis of effects resulting in corn crops? I don't know if you want to unmute and kind of add to that.
Or would you like to elaborate on your question?
Oh, I just saw Howard. Or Howard, maybe that was answered in the
I think in general,
it's a similar question. Yep. He thought it was the
yeah, in
similar.
general, it's just reduction of yield. Yeah.
Yeah, awesome. Does anyone else have any questions for Jose? Nope. All right. Well,
Thank you
thank
very
you
much.
so much. We really appreciate it.
Thank you.
All right. And I want to introduce our next speaker, Courtney. And Courtney is coming to us. She is a PhD candidate at the University of Wisconsin-Madison. She's in Dr. Leslie Howard's fruit crop pathology lab. And she's worked on a variety of fungal pathogens, primarily those affecting fruit crops. Courtney's first research project in 2018 utilized fungal morphology and phylogenetics to identify new fungal species of the genus Neophysium. I'll let you say that better, a little more clearly, with Dr. Martin Brewer at the University of Georgia. Following this, she desired field-based pathology research, which led her to Dr. Philip Brennan's lab. There, Courtney conducted a basal field study examining the infection period of Mononilla fructicola on peaches. So after graduating from University of Georgia, she knew that graduate school was the next step, which led her to the University of Wisconsin in Madison. Her passion for plant pathology is rooted in understanding fungal biology, and etiology to gauge the agricultural industry to be more sustainable or to aid the agricultural industry. So, Courtney, thank you so much. Courtney Meeks, thank you for joining us.
Yes, thank you all so much for spending your Monday evening with me. And Jose, great talk. We have some tar spot people in our department. So
Thank
it's
you.
always
Thank
nice
you.
to hear it from different perspectives, and always love a nice phylogenetic tree. So hopefully y'all can see my slides and everything.
Yes.
Yes. So thank you all for joining. If I sound a little funny, I just got back from being home over the weekend, so my southern accent is in full force today. But and I'm just so thankful to have been chosen as one of the scholarship recipients and to share with you my research that I'm doing with Dr. Leslie Holland, specifically assessing fungal disease management for cold climate wine grapes here in the Midwest. So I'm going to talk a little bit about cold climate wine grapes. I don't know everyone's familiarity with the industry, talk about diseases and management, talk about some of my field trials I've completed, my resistance research, which is the biggest thing I'm currently working on, and kind of moving forward as things are warming up, spring is sprung, and the grapes are alive, which means the fungi are alive. So I'll be out in the field. So cold climate wine grapes, they are so unique. They are actually the attributor to breeding these is Elmer Swanson. He was a dairy farmer, actually kind of towards the Wisconsin and Minnesota state line. And growing up, he's truly a citizen scientist. He was like, wow, we have these wild grapevines, but we can't grow wine grapes here or table grapes because it's so darn cold. So he decided to start breeding grapes in his backyard when he wasn't milking cows every 12 hours. And he created what we call an interspecific hybrid. So these are grapes that if you can think about your own family tree, you might be German or French Canadian, and you have all these different in your family tree of so many different maybe immigration contributions. Likewise, that's what he was doing. The initial cross was with Vitis vinifera, the European wine grapes we all know, the Chardonnays, Cabernet Sauvignon, but crossing them with the wild grapes he grew up with here in the Midwest, such as Vitis ripara and Vitis labrusca. And he would breed once and then breed again and just would kind of this web of different crosses. And the purpose was he wanted the fruit chemistry of a wine grape, because the wild grapes, if you've seen them out foraging, they're literally for the birds, super acidic, small fruit set. But he needed the cold hardiness that the native grapes had. And so this has resulted in a growing industry throughout the Midwest. And actually, the University of Minnesota is home to one of the best cold climate grape breeding programs. So if you ever have the chance to go to Soon Lee's lab, it's a great time. And we have some common cultivars here. So if you're out shopping for wine and you're interested, Marquette is a red grape and La Crescent's a white. Those are some of the original Swansons. And then two emerging are Petite Pearl, which is my favorite grape, mainly because it gets my favorite disease. But you see, this is the grape here, this beautiful tight red, this tight cluster, and then Itasca, which is a growing white cultivar. So these are the up and comers, if you will. So of course, as a plant pathologist, we have our holy grail, right? The disease triangle. Disease is not spontaneous. It is the culmination of three things. So first, a susceptible host has to be present, right? So we're growing these grapes here in Wisconsin and throughout the Midwest. The pathogen has to be present, and all the pathogens of the common fungal pathogens of grape are endemic to the Midwest. But the biggest thing is the environment has to be conducive. So you need the right humidity or heat in order for the pathogen to infect, grow, disperse. But in sometimes, especially in certain systems, to make the host even actually make the host susceptible, make it stressed. And when all of these things culminate into perfection, you have disease. So current management strategies, this is a beautiful image of powdery mildew. It's an Erisiphie nectar of on wine grapes. I try to get in as many beautiful diseased images as I can. But really, the root of management strategies is you have to know the enemy, right? You have to know pathogen biology. So understanding where's infection beginning, what's that primary inoculum, what environmental conditions actually favor this pathogen to complete its life cycle. And then, of course, how does the host interact with these pathogens? Is it very susceptible? Is it tolerant? Is it resistant? And so previous research kind of aids us a little bit here. So first, one unique thing about cold climate wine grapes is when Elmer Swanson was breeding, he wasn't just choosing the cold hardiness genes and using some gene technology to insert it into a wine grape. He was doing traditional breeding. So he not only was able to breed in cold hardiness, but he also bred in these natural disease tolerant and potential resistant genes that these wild grapes had just being in the environment for thousands of years with these pathogens. And so this is actually an area of research where here at UW-Madison, and they also did it over in Vermont, they were able to not spray a vineyard for 2 years and just see what would happen. And they were able to show that these cold climate grapes have different interactions with these fungal diseases. And they're not all the same in susceptibility. Vitis vinifera, domesticated in the Mediterranean, is very susceptible to every disease that we have here in the United States. And so, interesting enough, they were able to show that these cold climate grapes, of the 3 pathogens, most were pretty tolerant, if not resistant, to 2 of the 3 pathogens. So it shows promise for us to treat these grapes in a unique way. Sanitation methods are very big, so we're not spraying bleach on any vines. Instead, we're trying to reduce primary inoculum. We're trying to remove as much disease material before the season begins. This can be mummies. So if you're ever out in a vineyard in the winter, it's quite literally like a little hard raisin, and you'll sometimes, most often, see little pycnidia or pseudothecia actually on that grape. And that's just chilling, sitting there, waiting for that conducive environment, typically spring rains, warmer temperatures to actually start to release those ascospores and conidia. And pruning, removing any old tissue, just to kind of sanitize in a way your vineyard. Oh goodness, there we go. Ontogenic resistance is a big area of research, especially in Cornell around 2008. And so ontogenic resistance is age-related resistance on the host side. So we're able to understand that really for some of these pathogens, there's only certain windows at which infection is really a problem or where it's super susceptible. And so this helps vineyards to be more strategic in when they're applying their fungicides. And lastly, if you go out west, California, Oregon, UV light is a really big area of growing research. There's a lot of hurdles because it has to be at night. There's a lot of time, what kind of apparatus you can construct. But UV almost basically kind of sterilizing, if you will, the fungus without harming the actual grapevine. And this is really big for powdery mildew. But here in the Midwest, our current disease management strategies are driven by these European wine grapes, the Vitis vinifera, because we've only really been around the Grower Association in Wisconsin since 2008. So we're a very young industry. And so we're utilizing data and historical knowledge from the parental lines of these, but we're not exactly the same. We're a little bit unique. And you can see this here. Excuse me, in my research was kind of tailoring disease and understanding disease management. So if we think about the disease triangle, this is a more interpretive image here, but we have the same fungal pathogens. The same fungal pathogens are infecting cold climate interspecific hybrids, just as they are European wine grapes. But our environmental conditions, we are not in the Mediterranean. Some days I wish I was in the Mediterranean, but we are not. We have a cool, temperate climate, and our hosts are very different. And so we need to, in order to be sustainable, we need to understand disease in the Midwest. So understanding how these unique cultivars in the Midwest weather change the disease triangle. And really, there's a desire amongst growers and, of course, consumers as well. We want to be sustainable with what we're doing. And this all kind of culminates into what we call the disease toolbox, right? So it's this metaphorical, this metaphor of we don't have a bunch of hammers. We have a hammer, we have a screwdriver, we have a needle nose pliers. We have different tools that can address disease in different ways. So the first is cultural, so those sanitation methods, cleaning out your vineyard. How can you make the environment less conducive? Chemical's a big one, fungicides. I'll be talking about that for the rest of my talk. The host, understanding, okay, this cultivar I'm growing here, is it highly susceptible to downy mildew, which is Plasmaperia viticola? Because if so, I'm not going to put that near water because it needs a lot of water to sporulate, right? So understanding your host, and then biologicals. This is actually also another area I'll talk about, but this is a growing area of research of understanding how different microbes can be utilized to outcompete plant pathogens or induce systemic acquired resistance, or like inducing the plant's natural defense responses to kind of heighten it before infection. Okay, so the role of fungicides, right? So it's a big topic, and they're really important because that conducive environment, when the environment is so conducive, when it's super humid and it is just really hot, a lot of pathogens really love that. And so that life cycle is able to complete more rapidly. But not all fungicides are the same. So we actually specify them by a mode of action. So what that fungicide, is explicitly doing in the fungus. And it's a lock and key. It's very specific, but it's denoted by a group number. So all group 3s do the same thing. All group 11s do the same thing. And it kind of helps break down the jargon of the active ingredient here that I can't even pronounce. And also the trade name can change based on, where you're buying it from, the commodity it's listed for. And so this just kind of helps, all right, just give it to me simple. So it makes it easier for vineyards and for researchers. And fungicides are not exclusive to one commodity. So group 3s aren't just something you find in grape. You'll find it in corn. You'll find it in carrots. So these different groups are being used across commodities. But of course, fungicides are chemistries. They need to be treated with respect. So in order to even apply these at vineyards, of course, they're licensed. They're getting education opportunities. The growers that I work with are like top notch. They're like literally the best. I'm so thankful for them. And they understand these labels and that they're the law and how to basically be sustainable in the applications that they're applying. So my question first is, can we change fungicide programs? So if we've been treating these cold climate wine grapes like Vitis vinifera since 2008, but we know they're not the same, how can we change our fungicide spray programs to be better suited and tailored to these cold climate wine grapes? So this is where I did some spray trials in 2022 and 2023, where I was comparing, there's 2 separate studies. The first one is a conventional or like a calendar-based spray program, which is the traditional ways of spraying fungicides. You apply every 7 to 10 days, 10 to 14 days, and you write it on your calendar like it is a doctor's appointment and you don't miss it. And I was comparing that to a reduced spray program, so reducing the number of applications, as well as an organic spray program. Wisconsin's number 2 behind California for organic production. And so a lot of our growers are really interested in ways that they can utilize Organic Material Review Institute, or OMRI. It's a special logo on organically registered fungicides. And I was able to complete these studies at the ag research stations. We have several here in Wisconsin, but 2 have grapes, as well as collaborate with some fantastic commercial vineyards. And we were able to assess the 5 common fungal pathogens, and actually assess these via standard area diagrams, which I threw one up here really quick, because one really cool thing about science is you collaborate with the most brilliant individuals. I got to collaborate with a graphic artist who was able to show me exactly what 50, 60, 70, 80, 90, and 100% sporulation of this, it's actually an oomycete pathogen, would look like on a grape leaf for my ratings. And so it's just a really cool way to highlight how science and creativity can really come together to address needs for growers. So I'm going to share just a couple graphs here of these spray trials because I know I'm kind of limited on time. This first one is actually, it's been accepted for publication. And what this is showing you is across all 5 pathogens, the conventional is in red and the reduced is in blue. I realize it's not the best colors. I do apologize. But average disease here is on the y-axis, and this is weeks across the season up until harvest. 2% disease, really low. Now, granted, this was a low disease pressure year, but by and large, you can see that these are always overlapping. So we're actually seeing there's no difference in how these 2 treatments, reducing the number of fungicides, is actually, there's no difference in how it is performing in managing the 5 common fungal pathogens. This is actually the organic trial. Once again, height of 2%. Now, side note, it goes from 0 to 100. So very low disease pressure once again. The organic is a little more up and down, which is to be expected. Organic fungicides typically do not linger as long. Their effect is a little bit shorter. But even though you have these ups and downs, statistically, there's no difference in how they performed. And this really is giving growers a lot of confidence and able to kind of start transitioning and dipping their toe, if you will, in organic, because there really hasn't been a lot of information on organic for cold climate wine grapes. So a lot of growers, they don't want to take the risk themselves. So we were able to kind of show them, hey, like, here's what it would look like. Here's what we learned from it. And I'm really excited. We have several growers who, since we completed these studies in 2023, last year we got to sit down, Dr. Holland and I, who's my advisor, and chat with them. And this year they're going to be kind of really transitioning. And we're really excited to see our research in other fields. So now we get into the interaction between fungi and fungicides, right? Because when you're applying a fungicide, something's happening, right? And in 2021, when I first joined the lab, we sent some isolates off to Michigan of Recipients nectar, powdery mildew, and they all came back resistant for QOI fungicides, which I'll talk about in a moment. And this kind of, for our growers, they're like, oh, you only sent 6 samples. That's a small percentage of all of our vineyards. We want to know more about what fungicide resistance is looking like in our vineyards. So fungicide resistance, what is it? It sounds scary. I go back and forth, but fungicide resistance in simplicity is the loss of sensitivity by pathogenic fungi to fungicides. So a fungicide is no longer as efficient at controlling a fungus. It's a cosmopolitan issue. You can just Google fungicide resistance. Don't Google it. Don't Google anything because it's just going to make everything crazy. But it's something you'll see documented in corn and carrot. It's everywhere, even in the medical system. So you'll actually get a lot of reports from hospitals. And resistance develops in different ways. It's not all the same. The first is pointed. So you can have specific changes, specifically mutations that happen that no longer can that fungicide locking key connect. There's a disconnect. So you're starting to see the fungicide is no longer it's not actually controlling the fungus as well. The other is detoxification. So the fungus can use its efflux transporters to push out the fungicide. It sees it, it's like, nope, and it just increases the gene expression, and it's like it was never there. Which all this can lead to multi-drug resistance, which has been documented in some cases. And basically, a fungus, is exposed to a fungicide it's never even seen before, a group number it's never seen before, and the fungicide doesn't work. So this is a growing area of research in understanding what's happening and how can we help farmers, growers, and even in some cases, medical professionals make better decisions. Testing is twofold. You want to see the genetics, you want to see the genotype, but you also need to see the phenotype, the physical, what's physically happening. So typically you're doing some kind of genotyping. You're looking at a target gene. Where's the fungicide actually targeting? That's where most people start. Then they'll go on to looking at what other things are being expressed or decreased in expression. How are they actually interacting with the fungicides? This is that phenotypic, the physical happening. So sometimes it's field trials. It's seeing, I have this much black rot, which is my favorite fungal pathogen. It's Phyllachora amplacida. I have like 50%. If I apply this fungicide, do I see a reduction? Poison play assays, which basically that's here in the lab, and we grow fungi on media, and we just add the active ingredient at different concentrations, compare it to a control, and see if the fungal growth is reduced in size. If so, that means the fungicide is working, or if we're not seeing a change in the fungi's growth, meaning the fungicide is not working. And there's different levels of resistance. You have partial resistance, meaning there's a little bit of a contribution of this genotype to what's happening in our field trials or in our poison play assays. And then there's complete, meaning there's a strong correlation between this mutation or this that's happening genetically with what we're seeing in the field or on our plates. So the fungi of interest I'm working with here are two obligates as well. So powdery mildew, Recipients nectar, ascomycete, polycyclic disease, and the other one is, excuse me, I have a frog in my throat. The other one is downy mildew, which is Plasmodium viticola. It's an oomycete, so it's actually a water mold, also polycyclic. And so typically downy mildews on the bottom of the leaves, you have these beautiful, absolutely gorgeous sporangiophores with the sporangia, the zoospores, so it needs that wet environment. And I just could look at diseased grape leaves all day long. It's my favorite. But specifically, I'm looking at group 11, or the quinone outside inhibitor, QOI fungicides. So these are called your strobilurins. And this is just an image here of ATP synthase. So this is how the fungus is producing energy. And so you can see here, each one of these red pieces is a different group number of a fungicide. And it shows you how specific these fungicides are at their target. And so this is a high risk resistance development because it's a single site. It's targeting one thing. And it's specifically in the cytochrome B gene, and it's affecting the electron transport. So this fungicide is applied and the fungus can no longer produce energy. It can't produce energy. It dies. And like I mentioned, genetic resistance, the genotyping has been pinpointed to changes in the cytochrome B or CYP gene. So how do we go from the field to the lab? How do we even test these fungi? So first, once again, beautiful disease leaf. So this is a vineyard. I'll go out and you can see all of these are different colonies of Recipients. And so you want to find the most single circle because they typically are very radial in their growth. You don't want to collect multiple colonies because then you're not going to get clean results. So you find like one little clean circle, and we use quite literally a sticker. It's called a tough spot, so it's a tough sticker. And we put that on with forceps. We're very clean about it, as clean as we can be in a field. We remove that tough spot really carefully, only trying to get the actual fungus. We don't want any of the plant if possible. And we put that into a test tube and we throw it in the -20 freezer until we're ready to process. Once we're ready to process, which is typically after harvest, we use a Chelex DNA extraction. So once again, science, using other science and collaborating with people. Chelex is actually a compound used by people in, I'm blanking on the term, forensics. So to extract DNA from materials for different, cases they might be working on. So we were able to extract just the DNA off of that sticker. We're able to amplify that target gene since we know that is where the source of resistance is coming from, that CYP gene. We're using polymerase chain reactions. We send that off for sequencing. So we send it just across the street, and then they're able to get us the actual genetic code. We get it back, we pull it into our molecular processing, and we're specifically looking for the G143A mutation, which confers complete resistance. And basically, it's at the 143rd amino acid, a glycine becomes an alanine. So you're having, once again, that lock and key is no longer functioning. There you go. So current results, I'll be sharing some preliminary results from last year. This is just a quick little map here. So the Burgundy counties are counties that are vineyards that I had the privilege of going out and collecting fungal tissue from. The ones in salmon are ones that we're really trying to get to this year. There was, field season's crazy. We weren't able to collect last year. And so we've actually already, we've been able to talk to these vineyards, and we'll hopefully be getting out there here in a couple months. So just for downy mildew, the Plasmopyria species, we had 100 samples. Of these, 70 came back with the mutant allele, meaning genotypic resistance. The other 30 were the wild allele, or the sensitive, still sensitive to the fungicides. Interesting trends, mutant alleles were more likely found in commercial vineyards. Okay, so the genotype that would be resistant was more likely found in these commercial vineyards, which are using synthetic fungicides. On the flip side, wild alleles, the sensitive, still sensitive to fungicides, were more likely found in newer plantings, so a vineyard that is very young, or one that is a lower input vineyard, meaning either they only spray organic or they're spraying really they're spraying fewer times on average. Interesting enough, there was one vineyard here in Dane County. The oldest I sampled, these grapevines were gnarly. They were from the 1970s. I actually was able to detect both alleles. So really interesting just to kind of have a picture genetically of what the state looks like. But really moving forward, we're sampling again this year. I'm collaborating with the same vineyards. I'm doing a disease tour collection. And this year, because we know certain vineyards where we're more likely to find a sensitive or wild type, meaning the fungicide should still work, and then a resistant type, I'm able to go out to those and actually conduct bioassays this year. So we want to see, okay, we know what it is genetically, but what is actually happening with the actual fungicide? And so because it's an obligate, we can't just plate it. Instead, we have to bring in grape leaves and expose the fungicide and the fungus and see what is that actual interaction like. Powdery mildew has been a problem to get processed. There's just been a lot of troubleshooting with the actual in-lab diagnosis. Potentially, it could be heteroplasmy in the samples. That's what a lot of the papers kind of attribute. But all my downy mildew samples came back very clean. I was never detecting two alleles, or so having the mutant and the wild type in the same colony that I collected from. But that is a potential. I'm still trying to dig into the actual biology to see what's the probability of that and to what proportion would you expect it. And then my research is being used by Dr. Holland to actually make vineyard-specific recommendations. So what can vineyards do based on this research to be sustainable, to make the best decisions for their vineyard? And lastly, I want to share with you this really cool tool. So we talk about that conducive environment, right? The environment has to be ideal for the pathogen. And so one thing that we're trying to get vineyards to utilize more is disease modeling. So hopefully you're able to follow me here. I think so. So this is the Network for Environment and Weather Applications. We call it NEWA, and it's based out of Cornell. And so just like when we watch the weather in the morning, they're able to forecast the weather. Well, if we can pull that into a model that understands how the pathogen works, we should be able to predict disease. And that is what we're able to do here. So these are weather stations. I'm going to zoom out here. They are across, there's some over here in southeastern Minnesota. There's a couple over towards the west side. But there's tools for all different commodities. I'm going to scroll down here. If you're an apple or berry person, it's there for you. But for grape diseases. So over a cup of coffee, a grower can come sit down, click the nearest weather station, and they can see, based on the environmental conditions, is it favorable for the pathogen? If it's not, oh, well, then maybe I don't need to apply a fungicide. But for example, if it's red, well, maybe I need to be proactive. So it really helps vineyards make more strategic, sustainable decisions in when to apply these fungicides. And so we're trying to, a lot of our growers and vineyards are trying to slowly start implementing it. It's changing an entire mindset, but a lot have already started to adopt it and are seeing great results as far as managing disease. So with that, I want to acknowledge my advisor, Dr. Leslie Holland, and of course, our vineyard collaborators. Could not do my research without them. And my undergrad mentees, Preston and Maya, who have helped with deal with troubleshooting and everything else and just being good sports when out in, really hot, humid days. And of course, the funding that ensures that all this happens. And so with that, I'm happy to take any questions. I know it was a lot of information, and yeah, there's a lot of fun fungi in cold climate wine grapes.
Awesome. Thank you so much, Courtney. Oh my gosh, I love how passionate you are. You love finding the perfect disease leaves so you can study them. And it's fun to see a lot of the outcomes as well, and how you can really see the impact of your work so firsthand. That's got to be really really satisfying. I'm going to go backwards here on some of the questions, if that's okay.
That's fine.
So Howard was asking, what, if any, is the relationship between the natural bloom on grapes and the fungal surfaces, like the mildew you were showing us?
Yeah, I'm going to open the chat here. Let's see. It was relationship to the natural bloom on grapes and the fungal surfaces. This is a great question. So this is an area that me and my PI will go out to coffee, and we try to think of the best way to address these questions, because in some instances we know exactly how the fungus is getting into blooms. And oh goodness, this is really well documented for like monolinia species, right? It's like monolinia the mimic. It can mimic the pollen tube and actually infect. We don't know in grape. I tried a preliminary study last year that failed absolutely miserably, but graduate research. And I was actually spraying spores on clusters, bagging them, and then bringing them back to the lab, cutting them open, and staining them for fungi, and just trying to see, can we see how this is getting in? And there's a lot of troubleshooting with that method. I was able to see definitely a fungal tube growing down through the ovary, which was really cool. But yeah, it's a really unique relationship. A lot of research needs to happen to really help us time those fungicides. But in my field trials, one interesting thing is always around bloom, we see an uptick in disease foliarly. So we know there's some kind of unique relationship happening where the fungus is releasing a lot of spores around the exact same time the grapevines are blooming. So we're able to kind of see that through some of my data, but we really need to zoom in and understand exactly how it's getting in. That's the ultimate goal, if I could have a goal in any of my research.
Thank you.
Yeah. And then Howard, you also asked if is there any work being done on the U of M Arboretum's Frontenac crosses? And you talked about Gris, Blanc, and Noir. I don't know. I know Soon Lee's the new grape breeder there, and he just started, I believe, in August. I was able to meet him via Zoom a couple of times, so I don't know what his outlooks are for a lot of his research currently. I know that they're wanting to understand more of the genes that can be attributed to resistance. I think he's focusing on downy mildew because we do have such high humid environments that downy mildew loves. I love to see it, but he's going to be focusing on that a lot. So definitely Soonley is a great person to reach out to if you have specific questions. And I know he loves giving tours and bringing people out and talking about all things disease, grape breeding.
he'll even throw in wine. It's a great time.
Nice. Howard is a botanical illustrator. Howard, maybe you're going to have to start painting diseased grapes and study
That's yeah. My wife has 3 paintings submitted to the Flora Fauna Illustrata at the Arboretum. So of the 3 grapes, and we did tour the fields with the gentleman you're talking about, and not so much looking at the bloom is all part of the natural protective device on the grape itself, but it was so common to have different fungal infestations of the leaves. So the grape is being protected, but the leaves are getting destroyed. I'm sure there's all sorts of research being done. So just curious if that was part of yours.
It's something. So I actually came in as a master's student and then transitioned over to a PhD, and it was kind of trying to figure out what research I could accomplish in the amount of time. If I could go back, I have so many thoughts on understanding the differences in infection, understanding a little bit more that age-related ontogenic resistance on the host side. But it is true. The grapevine will sacrifice its leaves all day long as a way to just keep, okay, I'll sacrifice my leaves here, but really protective with its blooms. And it's a really interesting area, and I wish I had more time and my PI could, fund me longer. But
Great. Thank you.
Thank you. And then Peter had a question about peach canker. And he's a question about a cherry tree. He says, I have a cherry that let's see, I have a tree that the cherries no longer plums. Go ahead, Peter.
Anyway, I can probably explain it better than I wrote it. And the only reason I ask is you were doing, fruit stuff out of Georgia, and honestly, the University of Minnesota's extension service and stuff, if you ask them about peaches or cherries, they go, "Oh, you need to talk to some other university because we don't really know anything about that." So
okay.
I just curious if there's any research that's been done. I unfortunately the very first peach tree I purchased ended up having peach canker, and now I have peach canker. I'm probably on about my 5th tree.
Oh, gosh.
So I always have peach canker, and I don't really have any good alternative locations. So I don't know if you have any experience with that unpleasant fungal disease.
Yeah, peach canker. My advisor is a canker person. She loves canker diseases. So I'll actually ask her because
There you go.
I know cankers, but like she knows cankers. And
There you go.
she also is very, very familiar a lot more with some of the prunus. I'll definitely contact her, and I'll actually I'll put my email address in the chat.
Sure.
If you want to email me,
I
and
can.
that way I have a good contact for you. But yeah, she'll be happy to answer that.
Yeah, the other thing, I've got a cherry tree, and it's kind of taken over the whole tree. It didn't start out that way, but now the cherries, the flowers germinate, it starts producing a cherry,
Mm-hmm.
and it never swells.
It just stops.
It just stops and then kind of dries up and falls off. And it used to be only half of the tree. Now it's the entire tree, so I get no cherries at all.
That's the worst part.
Oh yeah, no, I mean I'm I've mentally threatened it with the chainsaw, but it hasn't helped. So
Is it a sweet cherry or is it a tart cherry?
it is a tart cherry. It's a Bailey cherry, which was developed in Canada. So it's a cold hardy tart cherry, but it's it's sweeter than some tart cherries. And in terms of the germination I get, I mean, I have the potential to get a lot of cherries if any of them would end up maturing at all. And I don't know what. And of course, I ask the people here, and they go, "Well, we don't know nothing about cherries.
Oh
Sorry."
goodness. Yeah.
So I don't know. I assume it's some kind of fungal
But you're not seeing any
critter.
kind of signs or symptoms. You're not seeing any kind of discoloration, any kind of fruiting body on the fruit.
No,
It's
no,
clean.
nothing on the fruit really at all, except maybe later in the season when, I've just left the stuff on the tree and then it looks like it's getting attacked by something later on. But
Yeah.
when it's in the process of not maturing, uh, it it I don't know, it takes a little bit of a red blush
Hmm.
uh, on the and all you see is like no actual fruit part. You just basically see the pit being
Yeah.
covered by a skin.
Interesting. I have a couple initial thoughts, but I don't feel comfortable making a recommendation yet. Because we do know that there's a lot of fungi that are latent. And when you start to see cherries ripening, so going through that blush, we know that they're all of a sudden they're producing anthocyanins and they're kind of starting to switch over into sweet mode. And a lot of fungi, that's a trigger for them to be like, it's a party, the sugar is here, let's come out of being latent, right? And so there could be a latent pathogen, but usually you see some kind of sign for that, right? You start to
Yeah.
see some kind of symptoms that could be like, hi, there's a fungus here. So my favorite, black rot, you see that. The latency window for black rot is like 2 months. So you get infections in June, you don't see those infections in the fruit until August. So it could potentially be something latent,
I mean,
but
these cherries, they mature and usually by early
mhm,
mid-July. I mean,
yeah,
if they were to normally, they come really early. Uh,
right.
you know,
Yeah, so and this one does have peach canker in it as well.
no,
Okay,
no,
okay.
just the peach tree's got
Just the
peach
peach tree
canker.
does. Okay.
I
I was
haven't
kind of
seen it.
wondering, I
I
was like,
mean,
wait a minute, is there
the tree is beautifully healthy. You look at it, dark green leaves and, and it blooms,
right.
prolifically. It's absolutely stunning.
Have
It's
you
the
done any kind
heat's
of
kind of took
soil
the, took,
have you done any
took,
kind of soil analysis to make sure that it's actually getting the nutrients it needs? Because there are some times that once the plant gets to the part of needing to actually put resources into the tree, or sorry, from the actually into the fruit, you can have where nutrient deficiencies can cause the plant to just, I can't support you, and just cut
okay.
it out.
Hmm. Okay.
So there are, and you should be, I don't know if Minnesota has a soil testing lab. José
Oh
might
yeah.
know.
Oh
Oh, they
yeah.
do? Okay.
They do. Yeah.
Okay. Yeah, so it could be maybe just see, I know with grapes, sometimes you would have uneven or bad fruit set because you're boron deficient, which is like a micronutrient. And so farmers end up going out and having to actually apply a foliar application of boron to kind of help with fruit set. So it could also be that. I'll dig into it a little bit more. But my first reaction would be latent infection or nutrient.
Okay. Thank you so much for allowing me to pick your brain.
Right. Yeah. No, I love getting pulled into, I'm in grape all the time. So any time to, except cranberry, my lab does a lot of cranberry stuff and I'm up to my eyeballs in cranberry. So anytime to venture into my roots of peach, I love. But yeah, peach canker is a one that I don't have the most familiarity with, but I can definitely ask Dr. Holland for sure.
Yeah, it's very nasty looking.
Yeah, field side manner, it's absolutely disgusting.
Yep, it absolutely is.
Uh, Yeah, I was just going to mention that Heather mentioned that she thought Elmer should be up for sainthood for developing cold climate grapes for wine. And I wanted to say thank you for that part of your presentation too. I thought that was really fascinating to know that somebody just old fashioned decided I want to grow grapes here to make wine. And
Yeah, it's the best part of cold climate wine grapes because of course we have wild grapes here, but I think cold climate wine grapes are the true like American grape. And it kind of shows you. I mean, I have a picture somewhere. He was elderly. He was not working. He was a dairy farmer. That was his job, but it shows that no matter where you are in life, you can accomplish amazing things and just being naturally curious. And he should be up for sainthood for I mean the whole industry would not have happened without him in his backyard. Just let me just see what happens right across these two and having the
It,
patience to do it as well.
yeah, because
I
that takes
remember when
right?
when I first learned there were cold climate grapes in Minnesota, and I was doing a tasting at a vineyard here, and I was like, "Oh my gosh, this is amazing." It just shocked me how good it was. But I mean I like sweet wine. But anyway, it was as good as anything you would get from Germany. So
Yes.
I find it interesting that that there was a bunch of other grapes that have been cold climate grapes that have been developed as table grapes.
Hmm.
Now they can't compete with table grapes in other climates, that are better. But for people that want to grow their own table grapes, there's some amazingly good table grapes
Fantastic.
that you can grow in these climates. You don't make wine out of them, but they're delicious. I ran into a school in northern Iowa that has a table grape vineyard that supplies their cafeteria.
That's fantastic.
It was so cool.
Yeah, so we have a small table grape vineyard that I scout. When I do my other diseases, I kind of just swing by and look at the table grapes, sample if needed for scientific purposes later in the year. But Minnesota has several cultivars or varieties that are becoming cultivars, and they are absolutely fantastic. Not only that, the leaves of table grapes are very different than wine grapes. They're very thick and they not rubbery, but they have a really unique feel. And so when I scout those, they even have a even more unique host fungi interaction than the wine grapes. They're even more tolerant and in some cases resistant to fungi, which makes them great for the backyard, right? You just kind of train it. Of course, you got to, put some effort into it. But we have several out at the West Madison Ag Station, which is just off campus, and we only spray them because there's a little bit left in the tank and we don't want to waste it. But you really don't have to. And the clusters are beautiful. The fruit is fantastic. And it's actually growing here in Wisconsin. We do know it's growing because some people came out and took some clippings and put them in their backyard. We're fairly certain. So it's definitely growing. And now if you go to the farmers markets, people are starting to sell Wisconsin table grapes, and people are so excited about it. So in some cases, a lot of people, the wine grapes are definitely increasing, but they've kind of plateaued. We're seeing wine consumption's not really increasing, especially amongst younger generations. So now people are like, well, I'm going to grow these table grapes. And that I think is really going to start taking off, especially once some of these newer varieties come out. And it's a fantastic, once again, don't know how someone had the vision to be like, I can breed these grapes. I can do this in my backyard and did it. And then got the support of the University of Minnesota to really scale it up and bring it even more to the public.
Awesome. Fascinating. Cutting edge.
Yeah, so go support Minnesota grapes this weekend if the weather's nice. Thank you all so much for listening and having such great questions and thoughts.
Thank you. Thank you so much. It's fascinating. Thank you, Jose. Thank you everyone for being here tonight.
Thank you so much for both of you. It's just great. We so enjoy these presentations.